Prior to carrying out sampling techniques, it is important that students understand why ecologists use sampling techniques. Ecologists monitor the size and distribution of populations which are food sources, or which are under threat from human activity, or which by their presence or absence indicate something about the environment in which they live. This film will consider the use of quadrats in both random and systematic sampling. When investigating an ecosystem, it is unlikely that every individual in a population can be counted. Pictures or wrapping paper or fabric could be used to illustrate the idea that you could take a sample and multiply it up to make an estimate of the number of organisms present in an area. In most situations, the distribution of organisms in a habitat is unlikely to be uniform. So what makes a good sample? Small samples taken together need to represent the whole population. A quadrat is a frame used for sampling an area. Quadrats can be any shape but are usually square. They can be any size, for example 10 cm by 10 cm for sampling lichens and mosses on a tree trunk. 50 cm by 50 cm is suitable for sampling playing fields. Some quadrats are marked off into smaller cells. You could even make your own simple quadrat from a coat hanger. Quadrats can be used to sample an area either randomly or systematically. Random sampling is done where the habitat is fairly uniform, or when data is needed to estimate the number of, for example, periwinkles in an area of rocky shore, or if a large area needs to be covered quickly. Some decisions need to be made about sampling. For example, how many samples? This should relate to the size of the area as a whole. How will we get a random sample? A common misunderstanding is how to place quadrats at random. Throwing a quadrat will not achieve a truly random sample. Instead, use some kind of random number generator. Lay two 20-metre lines or tape measures. Select a number randomly for each line. Then use the numbers as coordinates to place the quadrat randomly. Decisions also need to be made about how and what to count. What's in, what's out, for example. The key thing is that there is an agreed system and everyone sticks to it. Abundance can be measured by counting the number of cells in the quadrat that contain species. For example, here the abundance of barnacles is being measured, and then the abundance of bladderwrack. This kind of random sampling will not show up trends or changes across one area. There are systematic sampling techniques which will allow changes in a line within one area to be investigated. For example, using a line transect. A line transect can be used to show zonation, for example down a seashore, or across a woodland, or moving from a woodland to an open area. A line transect could also be used to investigate a linear area, for example the edge of a path. A rope or string is used to mark out the line. Quadrats are placed at regular intervals. Organisms' presence or abundance can be noted. Students may wish to look at abiotic factors affecting species distribution in a habitat. Factors such as moisture, light intensity, pH, or at the shore salinity of rock pools could be measured along a transect line and related to species distribution. By counting individual organisms or cells containing individuals, patterns of distribution or zonation can be studied. The ACFOR scale can be used to show the number of individuals, or percentage cover, against distance along a transect on kite diagrams. Following a visit to the rocky shore at King's Barns, one class of students used the ACFOR scale to produce these kite diagrams for a range of seashore populations.